This is a production of Cornell University.
Thank you, Jean-Luc, for the very kind introduction. Thank you all for being here, and thank you to my family and friends who are online with us today as well. I hope you're all enjoying the cassava snacks. Feel free to go back and get more. um, so yeah, I'm really excited to be here today to talk about my research titled Investigating the Genetic Basis of Trade-offs in Cassava Breeding for Improved Provitamin A and Dry Yield. In case you're not familiar with cassava, it's also known as yucca or manioc in different parts of the world. It's a major staple crop for millions of people originated in Latin America, but it's become a especially important food source in sub-Saharan Africa, where it provides 30 to 50% of the calories there. It's very drought resilient and low input crop, and it can be harvested when needed, so it's especially important for food security. It's high in carbohydrates, but generally is low in micronutrients. There's many delicious ways to prepare cassava in West Africa. Some of the favorites are fufu, gari, and eba. And yeah, that's cassava. Cassava-based diets have been associated with high rates of vitamin A deficiency. It affects 1 in 4 children in sub-Saharan Africa and can cause blindness, stunting, and susceptibility to infections. Vitamin A has to be consumed through the diet as provitamin A carotenoids like beta carotene, which are then cleaved in the human body to form vitamin A. And one of the ways that people have been working to improve the high rates of vitamin A deficiency are through biofortification of staple crops with provitamin A. And cassava has been a target of these efforts through the Harvest Plus project, which has been working to increase carotenoid content through traditional breeding in cassava to help combat vitamin A deficiency. A challenge with these efforts has been a trade-off between carotenoid content, TC, and dry matter, DM. Dry matter in cassava is mainly comprised of starch, so it's an important contributor to dry yield and also the textural properties of the cassava root. And so as breeders have been selecting for higher carotenoid contents, it's been harder to identify lines that have acceptable dry matter content as well. The genetic basis of this relationship between carotenoids and dry matter has been unclear. When I first started, the main hypothesis was that there was a genetic linkage between phytamine synthase 2, which is the major rate limiting enzyme in the carotenoid synthesis pathway, with another gene separately for dry matter, of which sucrose synthase was a top candidate. So it's thought that these two genes, separately but closely located together on the chromosome, were influencing this genetic relationship. And further, a wild integration segment from the wild relative Manihot was suppressing recombination in this region, which would make it harder to break that repulsion linkage. Other studies supported the hypothesis of pleiotropy, where PSY2 is affecting both carotenoids and dry matter directly. Studies that have expressed PSY2 as a transgene in cassava and other crops have seen shifts in dry matter and other aspects of primary plant metabolism, which suggests that carotenoid accumulation and synthesis itself can impact the carbohydrate content. Distinguishing between these genetic relationships is important for helping us understand the best breeding strategies to use to increase both carotenoid and dry matter content. If we have a genetic linkage, we can use targeted recombination or continued crossing with large enough population size. Eventually, the linkage can be broken. If pleiotropy is involved, we may need to use other loci or metabolic engineering strategies to break the relationship. As I started looking more into the relationship or the hypothesis of pleiotropy, I discovered that there were reports of carotenoid and carbohydrate trade-offs in many other crop species, including potato, sweet potato, butternut squash, citrus, banana, and others. And so I started to hypothesize what might be the interaction between these metabolites, which on the surface don't seem to be metabolically connected, but both carotenoids and starch are synthesized in the amyloplast in cassava roots. Amyloplast is mainly a starch synthesis and storage plastid. but carotenoids are stored in the membrane of the plastid. And both carotenoids and starch rely on the import of glucose 6-phosphate into the amyloplast for their carbon substrate. So 3 main hypotheses that came out of this literature review about the interaction between carotenoids and carbohydrates were precursor competition, where there's direct competition for glucose 6-phosphate entering the plastid, either towards carotenoid synthesis or starch synthesis. There may be physical interactions in the plastid where the carotenoids accumulating in the membrane are affecting the ability of the plastid to continue its starch synthesis role, or causing a shift in the plastid identity from an amyloplast to a chromoplast, which mainly stores carotenoids. And the third hypothesis is related to apocarotenoid signaling. So apocarotenoids are downstream of the carotenoid synthesis pathway, and they include the phytohormone abscisic acid, among other signaling molecules. And so some studies have suggested that apocarotenoid, and specifically abscisic acid, increase in abundance when increased flux is directed through the carotenoid synthesis pathway, which then results in stress responses in the plant. So this led to an addition of a mediated pleiotropy hypothesis, where phytoene synthase 2 is affecting dry matter via its relationship with carotenoids, and a physiological relationship between carotenoids and dry matter. Okay, so we approach this problem in 3 different ways. I'm going to present 3 projects. The first is looking at the genetic structure, specifically on chromosome 1 at the site of the hypothesized genetic linkage in the Glasioviae introgression. The second is looking at genome-wide relationship between the 2 traits with the bivariate genome-wide association study. And the third project is looking at physiological interactions, trying to understand the mechanism of the interactions between carotenoids and carbohydrates with gene metabolite network analysis. Okay, first, a little bit more background on the wild introgression that I mentioned on chromosome 1. So Manihot Glasioviae is a wild relative of cassava, Manihot esculenta. It was crossed with cassava in the 1930s to try to introgress disease resistance into the cassava breeding germplasm. And some of those progeny became important founders in the modern breeding germplasm. There's a large introgression segment on chromosome 1, about 10 megabase pair, that has been associated with important yield QTL, including root number and dry matter content. So it's been increasing in frequency in the breeding population, and has been an important source of variation for these traits. There's also suppressed meiotic recombination in the introgression, which complicates breeders' ability to break down the introgression and reduce the linkage drag associated with the favorable QTL. To investigate this, I sought to find map the chromosome 1 Glasioviae introgression. I designed Cas markers to tag the introgression haplotype, use those to generate a mapping population with more recombination in the introgression region. Did composite interval mapping for vigor and yield traits with the goal to identify introgression segments that could be selected for the QTL of interest or purged if they were carrying genetic load. So for this project, we generated a mapping population with the help of Ismail Rabi and Smith Ikpan at IATA Ibadan in Nigeria. And it has been a pleasure working with them and getting to travel to IATA to work with this project. So Smith was able to make these crosses between 41 parents that were heterozygous in the introgression region. And we planted this seedling nursery that was segregating for the introgression. I designed this panel of Cas markers that would tag Glasioviae specific alleles in the introgression region about 1 megabase pair apart. And so we used those to screen the large population of seedlings. And this is what the genotypes of those seedlings looked like across the introgression on chromosome 1, the 10 Cas markers here on the x-axis, and the 3,000 seedlings on the y-axis. And the Glasioviae alleles are in green for 1 copy or blue for 2 copies. We noticed that there was fewer homozygous introgressed lines than expected in a 1 to 2 to 1 ratio expected from crosses with heterozygous parents. And this segregation distortion was statistically significant across all 10 of the cassava markers, which indicated that the Glasgow-VI alleles might be having an impact on germination, early survival, or pollination success. We were still able to identify recombinants between every pair of markers and selected an optimized subset of 500 lines to advance to clonally replicated trials using Federal's exchange algorithm. And these 500 lines, we then genotyped genome-wide with DART-seq-LD markers. So into the composite interval mapping results, we found that the Glasgow-VI alleles are associated with lower vigor, both in the seedling stage and in the clonal stage, where the Glasgow-VI lines had slightly smaller stem diameter than non-introgressed lines. This also corresponded to a significant association with the breeder's rating of vigor on a categorical scale, of which stem diameter is a main component. And plant height is also a component that contributes to the breeder's assessment of vigor, but we did not find any significant associations with plant height. Surprisingly, we didn't find any significant effects of the Glasgow-VI alleles on dry matter or root number in this population, which were the main QTL that have been associated with the introgression. We did find a small association, small positive association with fresh fruit yield at the end of the introgression segment, but the effect size was fairly small. To investigate why we see suppressed recombination in the introgression region, I looked at genome alignment between the Esculenta and Glasgow-VI using a new Manihawk Glasgow-VI genome assembly recently assembled at the Salk Institute in Todd Michael's lab. Looking at the alignment between Glasgow-VI and Esculenta in the introgression region, we see there's fairly strong synteny between the genomes. There's no major structural variations, large inversions that would explain a recombination suppression in this region. There's several insertions and deletions, including this deletion, which is fairly large, but none that would explain the introgression-wide suppressed recombination. We also looked at the distribution of deleterious alleles in the Glasgow-VI introgression that might explain the reduced vigor and the reduced frequency of homozygous introgressed lines. So we used these putative deleterious alleles that were predicted by Evan Long, who looked at evolutionary rate as well as the SIFT score, sorting intolerant from tolerant, to predict whether mutations were likely deleterious. And so on chromosome 1 in the introgression region, there's a significant enrichment of putative deleterious alleles in the introgression region. And this difference was significant enrichment compared to the distribution of deleterious alleles genome-wide. So some key takeaways from this first project, we found that the Glasgow-VI alleles are associated with segregation distortion and that the introgression has negative additive effects on vigor and is enriched with putative deleterious alleles. The effects on dry matter and root number were not replicated, so therefore we can recommend breaking up this introgression. It's not clear why the benefit of the introgression in the population currently is not clear, so we can recommend breaking it up and hopefully can use those Glasgow-VI cassava marker panel to help prioritize recombinant individuals in selection. I'm going to give some acknowledgements after each project to thank the people who were involved in this project. So for this first project, the IITA cassava breeding unit team and IITA Abadan helped with the data collection as well as the maintenance of the field trials. So I'm grateful to all those who helped collect this data and worked on this project with me. Okay, so after this chapter brings us back to what did we learn about the genetic relationship between carotenoids and dry matter. We found that the introgression segment is probably not important to this relationship. There may be a linkage, but it's not necessarily related to the Glasgow-VI haplotype. So I became more and more convinced of a pleiotropic relationship and predicted that if there was such a physiological trade-off, as I hypothesized, then QTL genome-wide would be associated with both traits because of this physiological relationship, not just at PSY2. And so we sought to test if there were multiple jointly associated loci spread throughout the genome, which we would expect under this model. Further, to distinguish between direct pleiotropy and mediated pleiotropy, we looked at the statistical relationships and the pattern of these loci across the genome, where under a direct pleiotropy model, a SNP would be conditionally independent of carotenoids given dry matter, as well as conditionally independent of dry matter given carotenoids, because it has an independent effect on both traits directly, versus in the mediated model, the entire effect of the QTL on dry matter is mediated via its effect on carotenoids. So therefore, the genotype would be conditionally independent of dry matter given carotenoids, but not vice versa. To examine this, we looked across the yellow cassava breeding population at IATA Ibadan, Nigeria, which has been a population led by the breeder Dr. Elizabeth Parks there as part of the Harvest Plus project. We were able to genotype 378 accessions in that population that were still existent, and we genotyped them with DART-seq LD. There was a lot of historical field trial data that we were able to leverage for this project, where carotenoids are measured by eye-check spectrometer, which is a quick extraction method used to estimate total carotenoids that's more precise than the color rating that's been used previously. And we were able to use multiple locations and years. For the bivariate associations, we did a two-step analysis where first we calculated BLUPs from the field trial data across years and locations for carotenoids and dry matter. And then we used those deregressed BLUPs to test SNP effects throughout the genome, where we included PSY2, the PSY2 allele at the causal SNP as a covariate so that we could identify, so we could account for its major effect on the relationship between the traits, and also accounted for the rest of genome relationships with the genomic relationship matrix. So in this Manhattan plot, these p-values are from bivariate tests, so the peaks represent associations with both carotenoids and dry matter. And the location of the PSY2 allele that was used as a covariate is here in this orange dotted line. And so we found that there were multiple other QTL beyond the PSY2 locus that were associated with both carotenoids and dry matter, one on chromosome 1 and one on chromosome 10, which was new. We looked at the LD structure within the chromosome 1 associations to confirm that this QTL was separate and not in LD with the PSY2 locus, so confirmed it was separate. We then looked, so we set a strict Bonferroni threshold as well as a relaxed threshold to capture QTL across the genome that we may not be confident in individually as being significantly associated with both traits, but together can show about the pattern of their effect on the two traits throughout the genome. And so we looked at these 13 QTL peaks that passed the relaxed significance threshold and looked at the direction of their effects on the two traits. And most of them, 11 out of the 13, affected the traits in opposite directions, where it would have a beneficial effect on carotenoids and a deleterious effect on dry matter, vice versa. And this test we found was significant with the binomial test, where we can confirm that the QTL are significantly more likely to affect traits in opposite directions than random. In addition, we looked at the residual genetic covariance after accounting for PSY2 and the QTL SNPs. And in all of the association models, there was still significant negative genetic covariance. And in a model with all 13 of the QTL that we detected at the relaxed threshold, there was still significant negative genetic covariance between the traits. So this suggests that the relationship between carotenoids and dry matter is distributed throughout the genome, has a polygenic basis. And that supports this hypothesis of mediated pleiotropy, where any or most QTL in the genome that affect carotenoids would also affect dry matter. And using the linear model comparison, we found that this mediated relationship was most supported by the data. So some key takeaways from the second project, we found that the negative relationship between carotenoids and dry matter has a polygenic basis. We used multi-trait GWAS to help distinguish linkage from mediated pleiotropy. And we also found that the multivariate associations have higher statistical power than the univariate associations. And so I'd recommend to all of you to analyze your correlated traits together to benefit from that increased statistical power. Acknowledgements for this project. I want to thank Elizabeth and Gabby at IATA Ibadan for helping me lead this project, and the IATA cassava breeding team who collected the field trial data for these populations over the years. Moving into the third project, I wanted to understand how carotenoid accumulation in cassava is regulated beyond the PSY2 variant that has a major effect on carotenoids and has been the major QTL that comes out in most mapping studies for cassava carotenoids. I also wanted to understand what physiological mechanisms may connect carotenoid and starch metabolism in cassava. And so to look at this, we were able to get some cassava germplasm from the next-gen cassava crossing nursery in Hawaii, sent to us here to grow in the greenhouse in Ithaca. We looked at about 50 accessions that were both from Latin America, from Africa, and hybrids between them. And we found lots of beautiful genetic variation, phenotypic variation for carotenoid accumulation in the roots, including spatial variation, where some tissue layers within the root cross-sections have higher carotenoid accumulation than others within other tissue layers. And in particular, there's a fun phenotype that we call the egg yolk phenotype, when the inner layer is enriched with carotenoids, so it looks yellow compared to the outer layer. And this phenotype has been thought to be driven by a separate mechanism than PSY2. So it was important to investigate as potential novel regulators for carotenoid accumulation in cassava. So I predicted that if there is a physiological trade-off between carotenoids and starch, that physiological trade-off should exist across both genetic and spatial variation, where in addition to the correlation across genotypes, we would expect that there'd be lower dry matter in tissue layers that accumulate more carotenoids and vice versa. So to examine this, we did 2 years of greenhouse trials. The first, we selected 24 genotypes to represent the range in carotenoid variation and had 3 clonal replicates. And the second, we subsetted for 20 genotypes and harvested at 4 different time points between 6 and 9 months after planting to understand the developmental role and influence on carotenoid accumulation. So for each of these plants, we harvested 3 roots per plant, cut a central cross-section, and dissected them into inner and outer tissue layers. Those samples were each lyophilized and homogenized, and on each we did RNA sequencing, starch, glucose, and sucrose assays, and carotenoid composition analysis with UHPLC. Okay, if you do the math, that's a lot of samples. So what we found from this data, we found that the carotenoid and dry matter relationship is stronger in the inner tissue layer, which is this one compared to the outer tissue layer. And I've put pictures here to show how we separated the inner and outer tissue layers, which was not an exact dissection. It was an approximation by punching out the core tissue layer. We also found that the relationship increases in strength over time, where at 6 months after planting, there was essentially zero relationship between carotenoids and dry matter, and at 9 months after planting, there was a much stronger negative relationship. This pattern bore out statistically as well, with a significant interaction between carotenoids and time point on dry matter, even after accounting for genotype. Within total carotenoids, there's several carotenoids we were able to detect and separate with HPLC. And beta-carotene is the major carotenoid in cassava. It's about 80% of the total carotenoids. But we found a lot of the variation, especially between our African lines and our Latin American lines, was between accumulation of these cis-carotene precursors, phytamine, phytofluene, and zeta-carotene, versus these downstream xanthophylls, violxanthin, lutein. And so because we were curious about this variation, we tested the effect of these cis-carotenes versus the total other carotenoids, and found that it was actually the cis-carotenes really driving the relationship with dry matter, where in this model, only total cis-carotenes have a significant negative effect on dry matter, whereas the other carotenoids do not have a significant impact after accounting for the cis-carotenoids. And one thing that's important to point out about this as well, because we included genotype in this model, the significant effects of carotenoids on dry matter after accounting for the role of genotype supports that there is a physiological trade-off, where there's not just a genetic relationship across lines, but there's also a trade-off within the phenotypic variation within a genotype. Moving on to the transcriptomic data, we modeled gene expression with negative binomial generalized linear models, and we decomposed the expression into genetic and non-genetic factors. So we had genotype, of course, as our genetic factor. We had several non-genetic biological factors that we knew were likely affecting carotenoid variation, like the tissue layer, the time point. And then I did a surrogate variable analysis as well to account for latent technical variation, since RNA-seq studies are prone to being affected by technical variation. So from these models, we were able to extract genetic estimates of gene expression, which are expression blues, as well as estimates for expression driven by non-genetic factors, as well as overall expression estimates, which represent the batch-corrected gene expression. And for each of these components, I created a gene metabolite network with WGCNA. We found from this network, here I'm showing the overall network, including genotypic and non-genotypic variation, that the carotenoid metabolites, of which the major ones are in purple here in the purple diamonds, clustered in different co-expression modules than the carotenoid-related genes that were annotated as having specific functions within the carotenoid synthesis pathway, which were mainly in other modules. Of the genes that were strongly associated with carotenoids in that purple module, most of them had regulatory functions. They were the top associated genes were transcription factors related to stress response, signal transduction, or protein interactions. One gene I want to pull out that was in the list of differentially expressed genes associated with the egg yolk phenotype, or the difference between carotenoid accumulation in the inner and outer roots, was this early light-induced protein 1, which is known to bind to both chlorophyll and carotenoids. So that's, I think, an important candidate that could be followed up with to investigate its role in carotenoid regulation. We found that looking at the overall topological overlap between the carotenoids and the starch genes in the co-expression network, that the carotenoid genes were not particularly more strongly associated with the carbohydrate genes than expected by chance. So in this graph represents 100,000 draws of random genes in the transcriptomic data set that had similar overall connectivity to the carotenoid genes. And so the actual carotenoid-related gene set had similar topological overlap to the average of the random gene sets, which suggests that these carotenoid and starch gene sets are not co-regulated transcriptionally. We can pull out individual significant connections between the carotenoid and starch genes, even though overall they're not significantly overlapping, although we need to interpret these more cautiously since they're individual connections. But among the strongest connections between carotenoid and starch genes were pyruvate kinase, which is an important step in glycolysis, and also a strong negative connection between hexose kinase and PSY2. And so hexose kinase is involved here in the sugar conversion from sucrose to glucose, and pyruvate kinase is an important step in glycolysis. That is the branch point between the sugar going towards carotenoid or starch synthesis. So these may be important candidates to follow up on and are consistent with some of the hypotheses in the literature, but need functional validation. Okay, some key takeaways from this network analysis project. We found that carotenoid accumulation is predominantly regulated by genes outside of the main biosynthetic pathway in cassava roots, which suggests some possible regulators for follow-up studies to look at. We found that carotenoid and starch genes are not globally co-regulated at the transcriptional level. So despite their overall phenotypic correlation, the connection between them is not a transcriptional co-regulation. We did identify some of the strongest carotenoid and starch gene interactions, which do support the hypothesis of sugar partitioning being a central role either through direct precursor competition or through signaling, particularly with cis-carotene-derived signaling molecules, but those hypotheses need further testing. Okay, for acknowledgements for this third project, I would like to especially thank the undergraduate interns who have worked with me in collecting a lot of this data, including Kiara Gomez, who is a BTI RU intern last year, and Jiho Lee, who was in Gaurav Mogi's lab, as well as the many people who helped me conduct this lab work, which as you may know, Jean-Luc's lab group doesn't usually do wet lab work, so I relied a lot on the Sorrells lab to conduct these analyses, as well as Ted Tannhauser's lab at the USDA, who helped me with the carotenoid HPLC. Okay, so bringing it back to what drives the genetic relationship between carotenoids and dry matter, we started out with this model of genetic linkage affected by suppressed recombination and introgression segment. After all these analyses, my thought has moved to this new model where all of the chromosomes in the cassava genome are contributing small effects to this relationship, which is not as easy to deal with in breeding than breaking a linkage, but there is quite a wide distribution of SNP effect sizes across the genome on their relative effects on carotenoids and dry matter, of which the PSY2 locus is just one, which affects carotenoids positively and dry matter negatively. But there's also a lot of other variation we can draw upon, for which the SNPs affect traits in the same directions. And so moving forward with provitamin A biofortified cassava breeding, I think we still should be able to make progress for both traits using genomic selection with the use of careful selection indices, although it might take longer than if the traits weren't negatively correlated. And we've looked at some potential carotenoid and carbohydrate mechanisms of their interaction, And these are hopefully will provide candidates for future research. But at this point, there's still a lot we don't understand about the mechanism. Okay, as I wrap up, I just want to mention, in case I have accidentally convinced you that this all went very smoothly, my samples went through 4 freezer failure incidents during this project. We also spent months getting a biosafety permit to work with a transgenic line that our collaborators never sent us. I got sick with COVID in Nigeria. My hypothesis was wrong about the network analysis, and I didn't find the QTL that I was fine mapping. So I mentioned that all for the other graduate students in the room to encourage you to keep going, that it's hard and it's worth it. Finally, I would like to acknowledge my committee. Jean-Luc has been wonderful to work with, and I've really appreciated getting to work in his lab and learn from him, as well as he's encouraged me to pursue my own research interests that have extended beyond the main focus of the lab. And those extensions have been supported by Mike Gore, Lili, and Ismail Rabi, my committee members. I'm really grateful for their time and input. I'd also like to thank all the Jannink and Sorrel's research group lab mates who have given me feedback and helped me think through this project throughout the years and have helped me in the greenhouse, among other, among other things. I would like to thank my funding sources. I've been supported by a USDA NIFA predoctoral fellowship, as well as a Schmidtau-Novak grant from the School of Integrative Plant Sciences. I'd also like to thank Synapsys for being an excellent community of support over the last 5 years, many of whom in Synapsys have become my dear friends in my community, and I could not have done this without them. Of course, I also met Ricky through Synapsys, which has been one of the best parts of this journey in the last 5 years. And so I'd like to thank Ricky for all of the support that you've given me throughout this process. And finally, I'd like to thank both of our families who have been an incredible source of support for us going through this journey. And thank you to those who are listening online today and with us. I'd like to thank everyone for listening, and I'd be happy to take any questions.
Questions?
I'll ask questions. First off, just a wonderful presentation and research. So I was wondering, a lot of the carotenoids, of course, are going to be expressed in the leaves. Have you looked at the expression patterns of the genes in the leaves and the roots, and how correlated are they? And are they, essentially, is the constraint all coming from leaf expression, and you don't have a lot of freedom in the roots?
Yeah, so the question was about expression of carotenoid genes in leaves versus roots. I haven't personally looked at that, but other people have, and there's separate duplications of the genes that are expressed in the leaves and the roots. There is some overlap, but it's quite small, where the copy, like vitamin synthase 2 expressed in the roots, is not really strongly expressed in the leaves. And vitamin synthase 1 is not strongly expressed in the roots. I think this probably allowed for carotenoids in the cassava roots to take on possibly new functions. They're not contributing to photosynthesis or photoprotection in the roots since they're underground, obviously. They're not, they have an effect on preventing post-harvest deterioration, but it's not clear that they have any sort of fitness effect that's beneficial to the cassava plants as they accumulate in the roots. So I think that may have allowed them to possibly evolve other signaling uses.
We have one question online from Greg. Why has the wild integration you characterize been preserved in modern breeding lines if it is largely associated with detrimental effects?
That's a great question, and one that I've been thinking about too. Of course, when we started this project, we assumed that it was increasing in frequency in the breeding germplasm because of its beneficial effect on dry matter, which we were not able to replicate. I have a couple of hypotheses about why. It's possible that our study did not have enough power to detect a QTL that is real. It's also possible that the original identification of the QTL was affected by population structure, especially because in the original GWAS that identified that QTL, the relationship was accounted for with the genomic relationship matrix that excluded the Glasgow-VI specific alleles. So I think that if it is a confounding effect of population structure that affected the GWAS, it probably also affected the genomic selection predictions. And so that's something I'd like to look at further when I have time to subset that original GWAS study and see if it is confounded with population structure.
It's probably in your slides, and I just wasn't looking at the numbers carefully, but regarding the physiological trade-off partitioning possibility, could you give us some sense for the relative amounts of carbon that is in the carotenoids relative to the starch? Because my expectation is that it's vastly more in the starch. And so could you comment on how that might affect your hypothesis that it's a kind of a partitioning trade-off?
Yeah, absolutely. The question is about how much carbon goes into starch versus carotenoids. Starch uses a lot more carbon than carotenoids do. Most of carotenoids are C40 metabolites. Starch is long polymers of glucose that use much more carbon per molecule, as well as the total amount of starch in the roots is much, much higher in the micrograms per gram compared to the milligrams per gram compared to the carotenoids microgram per gram. So that is that has been one of our biggest skepticisms about the precursor competition hypothesis. And personally, I think that if that is the main trade-off, it's likely mediated not through a direct like one-for-one competition, but via signaling where glycolysis is upregulated to support carotenoid synthesis, and that is changing how the plant is partitioning its carbon between sugars and starch. We see increase in sugar content associated with carotenoids as well as increased fatty acid content. So I think it's not necessarily a one-for-one competition, but a transition into different prioritization of carbon metabolism. Any other questions?
Let's thank Seren again.
Please take some snacks with you.
This has been a production of Cornell University, on the web at cornell.edu.